What are microorganisms? Microorganisms are microscopic life forms that are invisible to the naked eye. Microorganisms can exist as single-celled organisms or as multicellular colonies. Microorganisms exist across three domains of life: bacteria, archaea, and eukaryota. To understand how small microorganisms are, let's compare their sizes to familiar objects. Viruses range from 20 to 300 nanometers, while bacteria are typically zero point 0.5 to 5 micrometers in size. For comparison, a human red blood cell is about 7 to 8 micrometers across. Larger microorganisms like amoebas can reach 200 to 300 micrometers, while a human hair is typically 50 to 100 micrometers in diameter. In summary, microorganisms are diverse microscopic life forms that exist across three domains and range from nanometers to hundreds of micrometers in size. In the 1670s, a Dutch businessman named Anton van Leeuwenhoek made one of the most significant discoveries in scientific history. Unlike the compound microscopes of his contemporaries, Leeuwenhoek crafted simple yet powerful single-lens microscopes. In 1676, while examining water samples under his microscopes, Leeuwenhoek observed what he called animalcules, tiny living creatures invisible to the naked eye. Leeuwenhoek documented his findings in detailed letters to the Royal Society of London. His claims of discovering tiny living creatures were initially met with skepticism. He included careful drawings of what he observed. Over time, as other scientists were able to replicate his observations, the scientific community accepted this revolutionary discovery. Leeuwenhoek's discovery of microorganisms fundamentally changed our understanding of biology. It revealed an entirely new world of life forms previously unknown to science. This discovery laid the foundation for modern microbiology and set the stage for later breakthroughs like Pasteur's germ theory and Koch's bacteriology. It expanded our understanding of biological diversity and challenged existing theories about life. Through his meticulous observations and detailed documentation, Leeuwenhoek opened the door to studying the invisible world of microorganisms, forever changing our understanding of life on Earth. Louis Pasteur, a French chemist and microbiologist, revolutionized our understanding of microorganisms in the mid-19th century. His work would establish fundamental principles that remain central to microbiology today. In Pasteur's time, many scientists believed in spontaneous generation, the idea that living organisms could arise spontaneously from non-living matter. It was commonly believed that maggots formed from rotting meat, mice from stored grain, and microbes from exposing broths to air. Pasteur was determined to challenge this long-held belief. In 1859, Pasteur designed his famous swan neck flask experiment to settle the debate. He prepared two flasks containing nutrient broth. One had a straight neck open to the air, while the other had a long curved neck shaped like a swan's neck. Both broths were sterilized by boiling. In the standard flask, microorganisms quickly grew in the broth, but in the swan neck flask, the broth remained sterile despite being exposed to air. Pasteur's ingenious design trapped microbes in the curved neck, preventing them from reaching the sterile broth below. Pasteur's experiment definitively disproved spontaneous generation. He famously declared, "Life only comes from life." His work established fundamental principles. Microorganisms come from other microorganisms, life doesn't spontaneously arise from non-living matter, and microbes are ubiquitous in our environment. These findings laid the groundwork for what would become known as the germ theory of disease. Building on his findings, Pasteur developed the germ theory of disease, the revolutionary understanding that specific microorganisms cause specific diseases. He identified microbes responsible for food spoilage and developed pasteurization to kill harmful bacteria. Pasteur connected specific microbes to specific diseases and created vaccines for deadly conditions like rabies and anthrax. These achievements fundamentally changed our approach to health and disease. Pasteur's germ theory revolutionized medicine and public health. In medical practice, it led to antiseptic surgical techniques, sterilization of medical tools, and hand washing protocols that dramatically reduced mortality rates. In public health, it sparked improvements in sanitation systems, clean water initiatives, and food safety regulations. Pasteur's work triggered a revolution in human health that continues to save millions of lives through disease prevention and treatment. Pasteur's brilliant experiments and germ theory fundamentally changed our understanding of disease and laid the foundation for modern microbiology and preventive medicine. Robert Koch was a German physician whose meticulous work in the 1880s revolutionized our understanding of infectious diseases. Koch established crucial links between specific microorganisms and diseases, developing methods that would become foundational to microbiology. Koch introduced revolutionary laboratory techniques that allowed him to isolate and study individual bacterial species. He pioneered solid culture media, enabling the growth of pure bacterial colonies. Until then, scientists struggled to isolate specific microbes. Koch also refined staining techniques and microscopy methods that made bacteria more visible and identifiable. These innovations gave Koch the tools to establish the relationship between specific bacteria and diseases. Koch's most enduring contribution was his set of postulates, criteria designed to prove that a specific microorganism causes a specific disease. These four postulates established a methodical framework that revolutionized the study of infectious diseases. First, the microorganism must be found in all cases of the disease and absent in healthy individuals. Second, the microorganism must be isolated from the diseased host and grown in pure culture. Third, the isolated microorganism must cause the disease when introduced into a healthy host. Finally, the organism must be re-isolated from the experimental host and identified as the same original organism. Koch applied his postulates to identify the causative agents of major diseases. His first significant discovery was Bacillus anthracis, the bacterium that causes anthrax. His most celebrated achievement came in 1882 when he identified Mycobacterium tuberculosis as the cause of tuberculosis, a disease that was responsible for 1 in 7 deaths at that time. For both diseases, Koch meticulously applied his postulates, establishing an undeniable link between the bacteria and the diseases they caused. Koch's methodical approach laid the foundation for modern microbiology and forever changed how we understand infectious diseases. His postulates remain a cornerstone of medical microbiology, establishing a framework for identifying disease-causing microorganisms that is still applied today. For his groundbreaking discoveries, Koch received the Nobel Prize in Medicine in 1905. His work solidified the connection between microorganisms and disease, validating germ theory and opening new avenues for treatment and prevention. Bacteria are single-celled prokaryotic organisms that lack a membrane-bound nucleus. Unlike eukaryotic cells, bacteria do not have a nucleus. Instead, their genetic material floats freely in the cytoplasm as a nucleoid. Bacteria come in diverse shapes. The 3 main shapes are cocci, which are spherical, bacilli, which are rod-shaped, and spirilla, which have a spiral form. Bacteria also form different arrangements. Diplococci appear in pairs, streptococci form chains, and staphylococci cluster together in grape-like formations. The bacterial cell has several key structural components. The outermost layer is the cell wall, which provides structural support and protection. Inside the cell wall is the cell membrane, which controls what enters and leaves the cell. The cytoplasm fills the cell interior and contains various molecules and structures. The genetic material, or DNA, exists as a nucleoid, not enclosed in a nuclear membrane. Ribosomes are small structures that synthesize proteins. Many bacteria also have flagella for movement. Bacteria show incredible diversity, with over 30,000 known species, adapted to virtually every environment on Earth. These microorganisms have adapted to extreme environments, from deep ocean hydrothermal vents to Arctic ice, desert rocks, the human gut, and even radioactive waste. Archaea represent a distinct domain of prokaryotic organisms, separate from bacteria despite their similar appearance. Unlike bacteria, archaea possess unique cell membranes with ether-linked lipids and genetic material that combines characteristics from both bacteria and eukaryotes. Most notably, archaea are known as extremophiles, thriving in environments that would be lethal to most other organisms. These remarkable organisms inhabit extreme environments like hot springs with temperatures up to 100 degrees Celsius, salt-saturated lakes, and deep-sea hydrothermal vents, where pressures are enormous and temperatures can exceed 120 degrees. The secret to their survival in these extreme conditions lies in their unique cell membrane structure. While bacterial membranes contain ester-linked lipids that break down in extreme conditions, archaeal membranes feature ether-linked lipids that remain stable in high temperatures, acidic environments, and high salt concentrations. Beyond their remarkable adaptations, archaea are of profound evolutionary significance. Many scientists believe archaea may represent some of Earth's oldest life forms, possibly emerging around 3.5 billion years ago. Their ability to survive in conditions similar to Earth's early environment makes them valuable for understanding how life evolved. Archaea appear to share a common ancestor with both bacteria and eukaryotes, making them a crucial piece in reconstructing the tree of life. Through their extremophile lifestyle, unique cellular structure, and evolutionary history, archaea demonstrate the remarkable diversity and adaptability of life on Earth. Protozoa are diverse, single-celled eukaryotes that often act as predators within microbial communities. These sophisticated organisms have complex cellular structures, varied methods of movement, predatory feeding behaviors, and play crucial roles in microbial food webs. The amoeba is a classic example of a protozoan. It has no fixed shape and moves using pseudopodia, temporary extensions of its cytoplasm. These pseudopodia not only help the amoeba move, but also capture food through a process called phagocytosis. The paramecium is a slipper-shaped protozoan covered in hair-like structures called cilia. These cilia beat in coordinated waves to propel the paramecium through water. They also help direct food particles toward its oral groove. Euglena is an interesting protozoan that has characteristics of both plants and animals. It has a long, whip-like flagellum for movement. The flagellum allows Euglena to move through water with a characteristic spinning motion. It has chloroplasts for photosynthesis, but can also feed on smaller organisms. Protozoa have evolved three main methods of locomotion: pseudopodia, cilia, and flagella. Pseudopodia are temporary extensions of cytoplasm used by amoebas. Cilia are short hair-like projections that beat in coordinated waves. Flagella are long, whip-like structures that create propulsion through water. Protozoa play crucial roles in microbial food webs. They are important consumers, feeding on bacteria, algae, and organic debris. They regulate bacterial populations, transfer energy to higher trophic levels, and help recycle nutrients in aquatic ecosystems. Their presence and diversity can also serve as indicators of ecosystem health. Fungi are essential microorganisms in ecosystems, acting as nature's recyclers. Microscopic fungi come in two main forms: yeasts, which are unicellular, and molds, which form filamentous structures. Molds form extensive networks called mycelium, composed of branching thread-like hyphae. This network structure allows fungi to efficiently explore and digest their environment. Fungi are nature's decomposers. They break down dead organic matter by releasing enzymes that digest complex compounds externally. The resulting simpler nutrients are then absorbed by the fungus while also becoming available in the ecosystem. Two of the most important microscopic fungi with practical applications are Saccharomyces cerevisiae, commonly known as baker's yeast, and Penicillium. Saccharomyces cerevisiae is used in baking, brewing, and winemaking. Its ability to ferment sugars produces carbon dioxide for bread rising and alcohol for beverages. Penicillium species are known for producing antibiotics like penicillin, which revolutionized medicine. They're also essential in creating the distinct flavors and appearances of blue cheeses like Roquefort and Stilton. Microscopic fungi are vital decomposers in ecosystems and valuable tools in human industry, from food production to medicine. Viruses occupy a unique position at the boundary between what we define as living and non-living entities. They share some characteristics with living organisms, such as containing genetic material and the ability to replicate. However, they lack others like cellular structure and metabolism. Let's examine the basic structure of a virus. A virus consists of genetic material, either DNA or RNA, surrounded by a protein coat called a capsid. Many viruses also have surface proteins that help them attach to and infect host cells. Unlike living organisms, viruses cannot reproduce on their own. They must infect a host cell and hijack its machinery to replicate. The virus first attaches to receptors on the host cell's surface. The virus then enters the cell where its genetic material is released. Using the host cell's machinery, the virus replicates its genetic material and produces viral proteins. These components assemble into new virus particles. Finally, the new viruses are released from the cell, often causing the host cell to rupture and die. Viruses come in a remarkable variety of shapes and sizes. Viruses range in size from about 20 nanometers to over 300 nanometers. Bacteriophages, which infect bacteria, have a distinctive structure with a head containing genetic material and tail fibers for attachment. Influenza viruses are roughly spherical with surface proteins that help them attach to respiratory cells. Pox viruses are among the largest and most complex viruses, with a brick-shaped structure and internal compartments. Despite their diverse structures, all viruses serve the same fundamental purpose: to deliver their genetic material into host cells and hijack cellular machinery for replication. Fermentation is a natural process where microorganisms transform food ingredients. During fermentation, microbes convert carbohydrates into alcohol, acids, and gases, creating unique flavors and textures. Let's zoom in on specific examples of microorganisms in food production. Yogurt is created when lactobacillus and streptococcus bacteria ferment milk. These bacteria convert milk sugar, called lactose, into lactic acid, causing the milk to thicken and develop its characteristic tangy flavor. Yogurt offers improved digestibility for lactose-intolerant individuals, probiotic benefits, extended shelf life, and unique texture. Next, let's examine how microorganisms create different types of cheese. Cheese making involves multiple microorganisms working together. Different cheese varieties use specific microbes. Blue cheese uses Penicillium roqueforti, cheddar relies on Lactococcus lactis, and brie contains Penicillium camemberti. The process begins with acid production by bacteria, followed by aging with specific microbes that develop the cheese's distinctive flavor and texture. Cheese offers a variety of flavors and textures, concentrated nutrients, long-term preservation, and enhanced protein digestibility. Now let's look at how yeast transforms flour into bread. Bread making relies on Saccharomyces cerevisiae, commonly known as baker's yeast. Yeast cells consume sugars in flour and produce carbon dioxide gas, causing the dough to rise. This fermentation process creates bread's light, airy texture, improves flavor complexity, enhances digestibility, and extends shelf life. Sauerkraut represents vegetable fermentation using lactic acid bacteria. Sauerkraut production relies on naturally occurring lactic acid bacteria present on cabbage leaves. Salt draws water from the cabbage, creating a brine where bacteria thrive. They convert sugars to lactic acid, naturally preserving the cabbage. This fermentation preserves vitamin C, extends shelf life, adds probiotic content, and enhances both flavor and digestibility. Finally, let's explore the surprising role of microorganisms in chocolate production. Chocolate production begins with fermentation of cacao beans by a complex community of microorganisms. Yeasts, bacteria, and fungi ferment the pulp surrounding cacao beans, creating heat and developing essential flavor compounds. This microbial activity develops chocolate flavor precursors, reduces bitterness, improves aroma complexity, and helps create chocolate's distinctive texture. Across all these examples, fermentation provides universal benefits to our food. These benefits include enhanced flavor, improved texture, extended shelf life, better digestibility, increased nutrient availability, and reduction of anti-nutrients. Microorganisms have revolutionized medicine, beginning with the discovery of antibiotics. In 1928, Alexander Fleming discovered penicillin, produced by the penicillium mold. This blue-green mold produces penicillin as a natural defense against bacteria. Penicillin works by inhibiting bacterial cell wall synthesis, causing the bacteria to rupture and die. Vaccines represent another critical medical application of microorganisms. Vaccines typically use weakened or killed microbes to stimulate our immune system without causing disease. The production of many vaccines involves growing microorganisms in controlled environments. These vaccinations trigger the formation of memory cells that provide long-term protection against pathogens. Genetically modified microorganisms have transformed medicine, particularly in the production of insulin for diabetics. Scientists insert human insulin genes into bacteria, turning them into living insulin factories. This was the first genetically engineered product approved by the FDA in 1982. The bacteria incorporate the human gene and begin producing human insulin protein. Probiotics represent another way we harness beneficial microbes for health. Probiotics are live beneficial bacteria and yeasts that support gut health and immune function. They're found in fermented foods like yogurt and kefir, as well as dietary supplements. These beneficial microorganisms help improve digestion, enhance immunity, and even produce certain vitamins in our gut. Microorganisms are workhorses in many industrial processes, offering sustainable alternatives to traditional chemical methods. They are utilized across various sectors, including enzyme production, biofuels, bioplastics, and biomining operations. Enzyme production is one of the most established applications of industrial microbiology. In detergent manufacturing, microbial enzymes target specific stains. Proteases break down proteins, lipases remove grease, amylases dissolve starch, and cellulases soften fabrics. In food processing, enzymes are equally valuable. Pectinases clarify fruit juices, lactase creates lactose-free dairy products, rennet is essential for cheese production, and invertase is used in confectionery. These enzymes are predominantly sourced from bacteria like Bacillus subtilis and fungi including Aspergillus niger and Trichoderma resi. Biofuel generation is another major application where microorganisms convert organic material into renewable energy sources. The process begins with biomass feedstock, which undergoes microbial conversion through various biochemical pathways. The end products are renewable biofuels that can replace or supplement conventional fossil fuels. Different microorganisms produce different biofuels. Algae are cultivated for biodiesel production. Yeast, like Saccharomyces cerevisiae, ferment sugars into bioethanol. Clostridium species produce biobutanol, and cyanobacteria can generate hydrogen. Bioplastic manufacturing harnesses microorganisms to create alternatives to conventional petroleum-based plastics. Traditional plastics are fossil fuel-based, non-biodegradable, contribute to persistent pollution, and have a high carbon footprint. In contrast, bioplastics are microbially produced, offer biodegradable options, reduce pollution, and generally have a lower carbon footprint. Common microbial bioplastics include polyhydroxyalkanoates, or PHA, produced by bacteria like Ralstonia eutropha, and polylactic acid, or PLA, which is derived from microbiologically fermented plant starch. Biomining is an innovative application where microorganisms extract valuable metals from ores through biooxidation and bioleaching processes. In the process, specialized bacteria oxidize sulfide minerals or produce acids that solubilize metal compounds, making them easier to extract. This results in the separation and recovery of valuable metals from their ores using microbial metabolism rather than harsh chemicals. Key microorganisms in biomining include Acidithiobacillus ferrooxidans for copper and gold extraction, Leptospirillum ferrooxidans for iron, and Sulfolobus species for uranium and zinc. Compared to conventional mining, biomining offers advantages including lower energy usage, reduced chemical pollution, and the ability to process low-grade ores that would otherwise be uneconomical. Microorganisms play crucial roles in sustainable agriculture. Let's first examine nitrogen-fixing bacteria, which are essential for plant nutrition. Nitrogen-fixing bacteria convert atmospheric nitrogen into a form that plants can use. These bacteria form root nodules with legume plants. These nodules house the bacteria that capture nitrogen from the air, making it available to the plant. This symbiotic relationship reduces the need for nitrogen fertilizers. Now let's look at mycorrhizal fungi, another important symbiotic relationship in agriculture. Mycorrhizal fungi form partnerships with over 90% of land plants. These fungi extend the plant's root system, dramatically improving nutrient absorption. The fungal hyphae act like root extensions, accessing nutrients and water that would otherwise be unavailable to the plant. This is especially important for phosphorus uptake. Microbial biopesticides offer environmentally friendly alternatives to chemical pesticides. These biopesticides use beneficial microorganisms to control agricultural pests naturally, reducing environmental impact and preserving beneficial insects. For example, Bacillus thuringiensis produces proteins toxic to specific insect pests while being harmless to humans and wildlife. These natural pesticides can be highly specific in their targets. Finally, let's explore how microorganisms drive the composting process, recycling nutrients from organic waste. Composting relies on diverse microorganisms to break down organic matter. Bacteria, fungi, and actinomycetes convert waste materials into valuable humus. Different microbes dominate at different composting stages. Thermophilic bacteria thrive during the hot phase, while fungi and actinomycetes complete the breakdown of complex compounds. These microbial applications demonstrate the immense potential of microorganisms in sustainable agriculture. Microorganisms serve as essential model organisms in scientific research due to their simplicity and rapid growth. These microbial models offer several advantages. They reproduce quickly, have simpler genetic structures, can be easily manipulated, and pose fewer ethical concerns than animal models. Escherichia coli, or E. coli, is one of the most widely used model bacteria in research. E. coli has applications in genetic engineering, protein production, DNA replication studies, and molecular cloning. Yeast, or Saccharomyces cerevisiae, is a single-celled fungus that serves as an excellent eukaryotic model organism. Yeast research has led to advances in understanding cell cycles, aging processes, protein interactions, and serves as a platform for drug screening. One of the most revolutionary discoveries derived from microbial research is CRISPR-Cas9 gene editing technology, which originated from bacterial immune systems. Bacteria use CRISPR systems as an adaptive immune defense against viral infections. These systems contain repeated DNA sequences that help recognize and cut foreign DNA. Scientists adapted this system into a precise gene editing tool that uses Cas9 protein to target and cut specific DNA sequences. Research with microorganisms has provided fundamental insights into four major areas of biology. Microbial studies have revealed critical information about cell function, including metabolic pathways, protein synthesis, and cell division processes. In genetics, microbes have helped us understand DNA replication, gene regulation mechanisms, and DNA repair systems. Evolutionary studies using microbes have illuminated adaptation mechanisms, horizontal gene transfer, and selective pressures that drive species development. Finally, microbial research provides insights into disease mechanisms, including infection processes, antibiotic resistance, and host pathogen interactions. These fundamental insights from microbial research continue to drive scientific discovery across biology, medicine, and biotechnology. Pathogenic microorganisms are microbes that cause disease in humans, animals, and plants. There are four main types of disease-causing microorganisms. Bacterial pathogens like Salmonella cause food poisoning with symptoms including diarrhea, fever, and abdominal pain. Viral pathogens such as the influenza virus cause respiratory infections with symptoms like fever, cough, and fatigue. Fungal pathogens like Candida albicans cause infections such as thrush and yeast infections in humans. Protozoan pathogens such as Plasmodium falciparum cause malaria with symptoms including cyclical fever, chills, and headache. Pathogens use different routes to transmit from one host to another. Airborne transmission occurs when pathogens travel through the air, like with influenza viruses. Foodborne pathogens like Salmonella spread through contaminated food. Direct contact transmission happens when pathogens transfer through touch, as with many cold viruses. Vector-borne diseases like malaria are carried by insects such as mosquitoes. Pathogens cause disease through three main mechanisms. First, toxin production occurs when pathogens release harmful chemicals that damage host cells and tissues, like the botulinum toxin produced by Clostridium botulinum. Second, tissue invasion happens when pathogens directly invade and damage host cells and tissues, as seen with HIV infecting immune cells. Third, immune response triggering occurs when pathogens cause excessive or harmful immune responses, as observed in severe cases of COVID-19 with dangerous inflammation. Understanding these pathogenic mechanisms is crucial for developing effective treatments and preventive measures against infectious diseases. Microorganisms are the primary cause of food spoilage, changing the taste, smell, and appearance of food. There are many types of microbial food spoilage. Molds form fuzzy colonies on bread. Bacteria create slime and off odors in meat. Yeasts can ferment sugars, creating gas and alcohol. Microorganisms need specific conditions to grow in food. Temperature between 20 and 40 degrees Celsius is ideal for most microbes. Adequate moisture with water activity above zero point 0.6 is essential. Foods rich in proteins, sugars, and minerals provide excellent nutrients for microbial growth. Food preservation methods work by creating conditions that inhibit microbial growth. Refrigeration keeps food at low temperatures where microbes grow slowly. Drying removes the moisture necessary for microbial metabolism. Salting creates osmotic pressure that dehydrates microbial cells. Canning uses heat to kill microbes and creates a sealed environment, preventing recontamination. Plant diseases caused by microorganisms have profound impacts on agriculture and natural ecosystems worldwide. The main culprits include fungi and water molds known as oomocetes, bacteria, and viruses. Each type attacks plants in different ways. These pathogens threaten crop production, food security, and can disrupt entire ecosystems. Let's examine potato blight, caused by the oomycete Phytophthora infestans, a fungus-like organism that devastated Ireland in the 1840s. Potato blight causes dark lesions on leaves, white fuzzy growth on the undersides, and devastating rot in potato tubers. This disease caused the Irish potato famine, resulting in 1 million deaths and massive emigration. Today, it still costs farmers about $5 billion annually. The pathogen spreads through water droplets, wind, and via infected potato tubers that are used as seed. Citrus greening is caused by the bacterium Candidatus liberibacter asiaticus, one of the most devastating diseases in citrus production. The disease is spread by tiny insects called Asian citrus psyllids, which transfer the bacteria as they feed on citrus trees. Symptoms include yellow mottling of leaves, misshapen bitter fruit, and eventually tree decline and death. There is no cure once a tree is infected. The economic impact has been devastating. In Florida alone, citrus production has declined by 70%, with losses of over $4.6 billion. The tobacco mosaic virus was the first virus ever discovered and remains one of the most studied plant pathogens. TMV causes a distinctive mosaic pattern on leaves, stunted plant growth, and significantly reduced crop yields. This virus is extremely stable and can remain infectious for years in dried plant material. It affects over 350 plant species beyond tobacco. Unlike many pathogens, TMV spreads primarily through physical contact between plants and via contaminated tools and hands during plant handling. Controlling plant diseases requires integrated approaches to minimize economic and environmental impacts. Developing resistant plant varieties is a cornerstone strategy. This includes traditional breeding and genetic engineering approaches to create plants that can withstand specific pathogens. Biological controls utilize beneficial microorganisms that naturally suppress pathogens. These include antagonistic fungi and bacteria that can protect plants without chemical inputs. Cultural practices like crop rotation, field sanitation, and optimized irrigation management help prevent disease buildup and spread in agricultural systems. Plant diseases have massive economic impacts globally. Annual crop losses exceed $220 billion. Despite investments in research and integrated management approaches, controlling plant diseases remains challenging. As climate change and global trade accelerate pathogen spread, understanding and managing plant diseases becomes increasingly critical for food security and ecosystem health worldwide. Microorganisms play a crucial role in breaking down various materials in our environment. These tiny organisms break down materials through specialized enzymes that convert complex molecules into simpler compounds they can absorb and use. Fungi are primary decomposers of wood using specialized enzymes like cellulases, hemicellulases, and lignin peroxidases to break down complex wood components. Wood decay takes different forms. Brown rot primarily attacks cellulose, white rot degrades both cellulose and lignin, while soft rot works more slowly in wet environments. Paper and natural fabrics are particularly vulnerable to microbial degradation because of their cellulose content. Cellulolytic bacteria and fungi produce cellulase enzymes that break the glycosidic bonds in cellulose, leading to deterioration of these materials. While most plastics were once thought to be non-biodegradable, researchers have discovered microorganisms capable of breaking down certain types of plastics. For example, the bacterium Ideonella sacchaensis produces an enzyme called PETase that can break the chemical bonds in polyethylene terephthalate, or PET plastic. Even metals are not immune to microbial degradation. Biofilms formed by bacteria on metal surfaces can accelerate corrosion significantly. This process, called microbially influenced corrosion, involves several types of bacteria, including sulfate-reducing and iron-oxidizing species, that create electrochemical cells on the metal surface. At the molecular level, microbial degradation relies on enzymes, specialized proteins that act as biological catalysts. These enzymes recognize specific chemical bonds, lower the activation energy needed to break them, and transform complex materials into simpler compounds that microbes can absorb and use. Microbial degradation has both negative impacts and beneficial applications. On the negative side, it can cause structural damage to buildings, deteriorate cultural artifacts, and lead to equipment failure in industrial settings. However, the same processes have positive applications, including the development of biodegradable materials, waste management solutions, and bioremediation of environmental pollutants. Understanding how microbes degrade materials helps us both prevent unwanted damage and harness these natural processes for sustainable solutions. From structural threats to innovative applications, microbial degradation represents a fundamental ecological process with far-reaching implications. Some microorganisms don't just survive in extreme environments. They thrive where most life cannot exist at all. Extremophiles are organisms specially adapted to survive in physically or geochemically extreme conditions that would be lethal to most life forms. Thermophiles thrive in extremely hot environments, like hot springs and hydrothermal vents, with optimal growth temperatures between 80 and 110 degrees Celsius. Their survival depends on heat-stable enzymes, specialized membrane lipids that remain fluid at high temperatures, and proteins that protect their DNA from heat damage. Psychrophiles are cold-loving microorganisms found in permanently cold environments like Arctic ice, deep ocean waters, and glaciers. They possess cold-active enzymes, antifreeze proteins that prevent ice crystal formation, and modified membrane lipids that maintain fluidity at low temperatures. Halophiles thrive in environments with extreme salinity, such as salt lakes, the Dead Sea, and salt flats, where salt concentrations can reach 15 to 30%. Their adaptations include maintaining high internal salt concentrations, special ion pumps, acidic proteins that function in high salt, and protective pigments that give many salt lakes their distinctive pink color. Acidophiles live in highly acidic environments with pH values between 0 and 3, like acid mine drainage, volcanic hot springs, and sulfur pools. They survive through highly impermeable cell membranes that keep acid out, reversed membrane potential, acid-stable proteins, and enhanced DNA repair mechanisms to counter acid damage. Barophiles, also called piezophiles, thrive under extreme pressure in the deep ocean trenches, where pressures exceed 380 atmospheres. Their adaptations include pressure-stable proteins, higher proportions of unsaturated fatty acids in membranes, and special pressure sensing systems that help regulate cellular processes. These incredible extremophiles demonstrate the remarkable adaptability of microorganisms to survive in conditions we once thought were incompatible with life. Their special adaptations not only allow them to survive, but actually thrive in these hostile environments, expanding our understanding of the limits of life. Microbiomes are complex communities of microorganisms that live together in a specific environment. The human microbiome consists of trillions of microorganisms that inhabit our bodies. These microbes are found in various locations, including our skin, gut, and mouth. Microbiomes serve many critical functions. They aid in digestion and nutrient absorption, help develop our immune system, protect us from pathogens, and even influence our mental health and mood. The gut microbiome is particularly important. It's our most diverse microbial community, with over 1,000 different species. These microorganisms are essential for breaking down complex carbohydrates that our body cannot digest on its own. Beyond humans, microbiomes are essential components of all ecosystems. They contribute to soil fertility and decomposition, water purification, and nutrient cycling. In fact, microbial communities form the foundation of all ecosystem processes. As we continue to study microbiomes, we're discovering just how important these microscopic communities are to all life on Earth. Microorganisms are vital engineers of Earth's ecosystems, performing functions essential for all life on our planet. Microorganisms are the primary drivers of essential nutrient cycles on Earth, including the carbon, nitrogen, and phosphorus cycles. They decompose organic matter and transform elements into forms that can be used by plants and animals, making them essential for ecosystem function. Microorganisms form the foundation of food webs in many environments, especially in aquatic ecosystems and soil habitats. As primary producers and decomposers, they serve as essential food sources for larger organisms, supporting entire ecosystems. Microorganisms are essential for maintaining environmental quality in soils, water systems, and the atmosphere. In soil, they decompose organic matter, release nutrients, and improve soil structure and fertility. In water systems, they filter and purify water by breaking down contaminants and cycling nutrients. In the atmosphere, they contribute to carbon dioxide fixation, oxygen production, and methane processing, helping maintain atmospheric balance. Without microorganisms, life as we know it would not be possible on Earth. They drive essential biogeochemical cycles, support food webs and biodiversity, maintain ecosystem health and resilience, and ultimately enable all other life forms to exist.